Hi everyone, so this is Mrs. Brady and this is the structure of DNA video notes. You should have your note-taking guide there in front of you and fill in as I go through the slides. All right, I've highlighted in yellow what you're filling in onto your notes there on your note-taking guide. So DNA is often called the blueprint for life. Essentially, a blueprint is the instructions to make something, and that's what DNA is. DNA is the instructions for making you, because it makes all the proteins inside of the cell, and then those proteins go to making you. If you aren't done filling this in, you might want to pause it because I'm going to slip ahead to the next slide here in just a moment. Go ahead and pause it if you need to finish filling it in. So Watson and Crick were the first two scientists to build the model of DNA. Notice though, they didn't discover DNA, they just built the first model of it. And this was about in the 1950s, so it wasn't that long ago that the structure of DNA was determined by James Watson and Francis Crick. There were other scientists involved in this discovery, but they're the two that are generally credited with discovering DNA, or sorry, not discovering it, but building the first model of it. Again, I'm going to move to the next slide. If you've not finished, go ahead and pause it. So next we're going to look at DNA and chromosomes. I think on your notes it says function of DNA. So our genes are on our chromosomes. Remember, we have 23 pairs of chromosomes for a total of 46 chromosomes in each and every one of our cells. And chromosomes are made up of a chemical called DNA. So over here on the right, we've got a picture of a cell. And inside that cell, we have 23 pairs of these X's, or 46 total. And then this is DNA here, this ladder, this twisted ladder. This twisted ladder is what makes up all of those chromosomes. So the function of DNA. A gene is a section of DNA that codes for a protein. And a protein, you are made up of thousands of different proteins that do everything from triggering an antibody response in your body to an allergy, like to milk, to estrogen and testosterone are hormones, to stomach acid that's a hormone, that's a protein. So you are made up of thousands of different proteins, and the code to make all of those proteins is actually found in your DNA. Each unique gene has a unique sequence of bases. So your color of hair, your skin color, the fact that you are right-handed or that you have something called a hitchhiker's thumb is all due to a series of bases that make you you. The unique series of bases codes for a unique protein, and those unique proteins then give us all of our unique characteristics. I'm going to move this to the next slide, so if you need to, if you're not done writing, go ahead and hit pause. So next we're going to get into the structure of DNA, specifically the shape here. So DNA is what we call a polymer because it's made up of smaller parts. And the basic shape of that polymer is called a twisted ladder. Some people say it almost looks like a zipper as well because there's teeth in the middle of a zipper that go together just like there's bases in the middle of a DNA molecule that goes together. But the actual term of the shape for DNA is called a double helix, which means there are two helical structures kind of wrapped around one another. So this is the actual name of the shape, is a double helix. It's like a twisted ladder or like a zipper. The actual name, though, is a double helix. Again, I'm going to go on to the next slide. If you're not ready, hit. So the double helix has two strands of DNA twisted around one another. So you can see that there's a green strand here twisted around the black strand. So DNA double helix, double because there's two strands that are twisted around one another. That's why we call it a double helix. You can see the black strand here and the green strand here. We're going to zoom in next into one of these strands. So here we have kind of some shapes to represent some different parts of DNA. The backbone of the molecule is alternating phosphates and a deoxyribose sugar. So the phosphate here is this yellow circle, and then the sugar is this blue-green, kind of an aqua-colored five-sided shape here. Kind of looks like a house. So that's our sugar. So it's phosphate, sugar, phosphate, sugar, phosphate, sugar. And that's what we call the backbone of DNA. If you think of DNA as looking like a ladder, this would be like the handrails of that ladder. So the backbone of DNA is phosphate followed by sugar, phosphate, sugar, phosphate, sugar. And the name of that sugar is called deoxyribose. And then the steps of the ladder that connect the two sides of the ladder are the bases, and they're called nitrogenous bases because their structures both have nitrogen in them. So the backbone or the handrails of this structure is a phosphate sugar, phosphate sugar, phosphate sugar. And then over here, where the little pink arrows are and the little yellow arrows are, those are the bases, and we call them nitrogenous bases. I'm going to move forward to the next slide, so if you're not ready yet, go ahead and pause the video. So nucleotides, that's the building block of DNA. And a nucleotide is made up of one sugar. Let's see if I can draw. One sugar right here, and it's made up of one phosphate group right here, and it's made up of one base right here. And that's what we call a nucleotide, and that's the building block of DNA. It is one phosphate, one sugar, and one base. So continuing on, one strand of DNA is a polymer of nucleotides here. And one strand of DNA has many millions of nucleotides. So your DNA strand here is millions of these nucleotides hooked up together. Notice we're only dealing with one side here. Remember, there's a whole nother side over here on this side to make that ladder-like structure. I'm going to move on to the next slide. If you're not ready, please hit pause. So in the middle, there are four nitrogenous bases. The first one is called cytosine, and it is usually found with the letter C. The next base is thymine, and it's abbreviated with the T. The third base is adenine, and its abbreviation is an A. And the last base is guanine, and its abbreviation is a G. So these are the four bases that make up your DNA, A, T, C, and G. And together, these four bases then code for all of your information. It gives you all the instructions to make you with all of these four letters, these four bases, A, T, C, and G. I'm going to move on to the next slide. If you're not ready, please hit pause. And there are two kinds of bases. The first type, oh, you're not writing any of this, by the way, just kind of listen for a second. There's two kinds of bases in DNA. The first ones are called pyrimidines, and that's because there's just one ring structure here for this and one ring structure here. So pyrimidines have just one ring. However, purines, on the other hand, have two rings. There's this ring right here. Let me see if I can draw. So there's this ring right here, and then there's this ring right here, and that's a purine. Thymine and cytosine are pyrimidines because, again, they just have one ring, one carbon ring right here that makes this five-sided structure. And then adenine and guanine are purines because they have two carbon rings. Again, you can see one ring right here and the second ring right here, and that's adenine. And then for guanine, we've got one ring here and one ring here. So back to your notes now. Remember that DNA has two strands that fit together, something like a zipper, and the teeth of that zipper, or the steps of that ladder, are in the middle. But why do they stick together here in the middle, right? And that's because of something called hydrogen bonds. Again, now you're starting to write again where we left off in the notes. So hydrogen bonds are what connects bases together in the middle of the DNA strand. Hydrogen bonds themselves are pretty weak, but because there's millions of hydrogen bonds, it's actually a pretty strong and stable molecule. When making hydrogen bonds, cytosine always pairs up with guanine. So C always pairs with G, so a purine pairs with a pyrimidine. And adenine always pairs up with thymine, so A goes to T. So C to G and A to T. If you can't remember what pairs, I always ask myself, well, where do I go to school? I go to school at CG, at A to T, CG, G to C. So just ask yourself, where do you go to school? And then you can remember what two bases pair together at CG. If you're not done writing, go ahead and pause it. I'm going to move forward to the next slide. We call this something called Chargaff's rule, that A always goes with T and C always goes to G. That's called Chargaff's rule. To crack the genetic code found in DNA, well, we have to look at the sequence of bases. That's what's super important here. And the bases are actually arranged in triplets, and we call those triplets codon. So down here, we have got 5 codons. I've got AGG, that's one codon. CTC, that's one codon. AAG, that's another codon. TCC, that's a codon. So each one of these is a codon, okay? It's a set of 3 bases. If you're not quite yet done, go ahead and pause because I'm going to move forward. In DNA, though, remember, it's double-stranded, right? So we have a sequence of bases then that pairs. Notice A pairs with T, right? So we have that one. G pairs with C. G pairs with C. C pairs with G. So we can know what the opposite strand is because we know what pairs, right? A always goes with T, C always goes with G. So we know exactly what the second sequence would be because we know A pairs with T and C pairs with G. You should be able to fill this in pretty easily. If you're not done quite yet, go ahead and pause it. I'm going to move forward to the last slide. So DNA by the numbers. And well, each cell has about 2 meters of DNA, and the average human has 75 trillion cells, which means the average human adult has enough DNA to go from the Earth to the Sun 400 times. That's how much DNA is in an average adult body. How in the world, though, I can't see it with my naked eye, how in the world does that happen? Well, it's because it has a diameter that is super, super tiny, right? Very, very small fraction of a size. And so that's why they're able to jam that much DNA into our cells. All right, guys, now you're going to go ahead and start practicing some of what you've known. You want to make sure that you tape these notes into your notebook because you're going to need them for a reference. Thank you.